Yeah, so good afternoon to all the registered participants for this course. If you are online, you can share your question with us. The participants which are not here, they can also share the question in the chat box. Otherwise, there is always scope to raise your question in the forum, which link you must be having, and there also you can share the questions. So far, I think we have covered 6 sessions, rather 6 modules, and that's so I will quickly revisit what we have discussed so far. Yeah, so these are the 6 modules we have. This is live so far. First one was the introduction to bioprocess engineering, where we discussed the overview of bioprocess engineering and what are the scopes. Then next, the first thing we discuss the metabolic stoichiometry. This is very much relevant for the media design as well as for the prediction of other byproducts and metabolites which are formed. Then we discuss how this stoichiometry can be linked with energetics, particularly in a simple way. We discuss in terms of degree of reduction or electron balance. So rather than material balance, one can also do the electron balance. The next topic was what are those biological agents which are relevant in bioprocess engineering to convert a substrate to product. So it includes enzymes and microbes. In addition, there are also mammalian cells and plant cells, but mostly this microbial bioprocessing is much more common. So we started this module 3 with enzyme kinetics. So there we discuss particularly this Michaelis-Menten kinetics, derivation of Michaelis-Menten kinetics and how to predict the kinetic parameter, particularly Km and Vmax. And in addition, we discuss what are the physical significance. Then we discuss how to calculate those kinetic parameter by simple double reciprocal plotting. And further, we discuss many times because the enzymes are biochemical catalysts. So we need to stop those reactions. One option is to go at the molecular biology level to stop the synthesis of those particular protein, which is acting as enzyme. That is a time-taking process. So other option is to add some inhibitor. So what we discussed the inhibition kinetics. So particularly in detail, we discuss this competitive, non-competitive, and uncompetitive. And further, as we move further, we discuss in fact this allosteric inhibition and substrate inhibition, because many times this high substrate concentration can be inhibitor. Not only, of course, it is inhibitor for the microbes, but it can also act as inhibitor for the enzyme kinetics. Then in the next module, once enzyme kinetics we discuss, so what is the issue with enzyme is how to reuse it, because it's highly expensive, and using free enzyme, at least practically, it is not economically feasible. So one option is to go for the immobilized enzymes. So those part we discuss in detail in module 4. The first we discuss different techniques for the immobilization. And once you are immobilizing means you are adding additional resistance. So what are those resistance? The first resistance come from the immobilization technique. The immobilization technique may have effect on both kinetic parameters Km and Vmax. And further, once it is immobilized, now it is going to affect the substrate concentration. So if you remember this Michaelis-Menten kinetics, it says V equals Vmax S upon, it says V equals Vmax S upon Km plus S. So Vmax and S are affected by the immobilization technique. Vmax and Km are affected by immobilization technique. Now the substrate concentration is also affected by the size or the porosity or permeability of that solid matrix. So now it is in solid matrix is imparting both external as well as internal mass transfer resistance. And internal mass transfer resistance. In a simple way, you can understand the concentration in the bulk was CSB. Then after external mass transfer, basically it follows a simple film theory. Concentration becomes CS. And now due to diffusion limitation, or you can say intraparticle diffusion, the concentration will further decrease to Ca. Now, how much decrease is there from CS to CSP or Ca? It depends on the effectiveness factor. If its value is 1, it means almost there is no mass transfer resistance. But in that way, one can analyze what is the effect of mass transfer resistance. Then finally, we move to this microbial kinetics. So there we have mostly discussed this unstructured and structured kinetic model, mostly focused to the monad kinetics, and further this along with microbial growth, how it is microbial growth is linked with substrate utilization and also for the growth associated and primary metabolites as well as for the secondary metabolites. Those parts were discussed. Then the last module, the 6th module was kinetics of substrate utilization, product formation, and biomass production. This was particularly discussed in different mode of operation, the batch culture, the batch culture, and as well as the continuous culture. And in continuous culture, we further discussed if continuous culture with the cell recycling. In addition to having the free or suspended microbes, we also discussed using this free enzyme or immobilized enzyme system for all 3 modes of bioreactor operation. Further, apart from continuous state time, we also discussed the plug flow bioreactor where there is no lateral mixing. So these were the 6 modules which has been discussed so far. And now next 6 module will include, the first one is 7th will be design and analysis of bioreactor. So you will see this, the 6th and 7th modules are more or less this interlinked. So there will be focusing more on the actual calculation part. Then once you discuss your enzyme, you discuss your microbial kinetic, then next comes the heat transfer and mass transfer process in bioprocess. So we'll be starting with the mass transfer, that is particularly much more relevant in the aeration and agitation. So we'll be mostly discussing on this aeration and agitation part. The next comes the heat transfer. So there we'll be discussing the mechanism of heat transfer, and followed by the more focus will be on the sterilization of both liquid as well as the air, which are relevant for the bioprocess. Then once it is done, then next comes bioprocess instrumentation and control. In order to control, we need to measure those parameters, and for measurement, we require the instrumentation. So we will be briefly discussing measurement of various parameters, how it is done, and then how these information or data can be used for the control purpose. Then second last module, it will mostly focusing on the downstream processing. Once the upstream is done, then how to recover and purify your desired metabolites. So different methods, starting with the separation of the cells, and then recovering your desired metabolite and purifying it. Those things will be discussed. And in the last week, we will be discussing bioprocess economics. Here also some part of downstream processing will be discussed along with the bioprocess economics. And in between, in the module 9, we'll also discuss the scale-up part, because it is very important in the bioprocess engineering. Whatever product or process you have developed at lab scale, that need to be translated at the commercial scale. So for that, what are the criteria which are used to explore the scale up, and from there how to choose which criteria is best suited. So those things will be discussed. Now I will open for the questions, then we'll discuss further. So the participants which are online, they can ask their questions. Otherwise, you can share your question in the chat box also. So I see a couple of participants here. So if you have any query, maybe related to this course or the whatever course content which has been discussed so far, if you have any suggestion also, that also you can please share. So as I can see a couple of participants. So do you have any query? I hope I'm audible to you. So what I can share, I can discuss some of the assignment questions. I hope my screen is visible. So for week 1, that it was introduction to bioprocess engineering. So for practice problem, can you share more question? Yeah, that's true. So for the practice problem, what particularly what I am doing, I am already discussing a couple of examples in my lecture slide itself. And in addition, in the assignment, you will be having some numerical problem as well. And in the numerical problem also, in the assignments, you are going to have some numerical problems. In addition, if you still have any Yeah, that's a good question. So are the problem presented in the video and assignment enough for the preparation of final exam? Yeah, the answer is yes, I think they are sufficient because the problems are based on the concept. And if those concepts are okay, then you will be able to solve problem which will be there in the final exam as well. But yeah, data may vary, and but however, the concept will remain same. And in addition, if you face any difficulty in solving any numerical problem from any other source, that also you can share with us and we'll try to provide the solution for that also. Thank you. Thank you so much. Is there any other query? Yeah, graphs, if it is needed, in the exam, typically the question will not be based on that where graph paper is required. If it is there, then the slope and this intercept value will be provided to you. Otherwise, mostly the graph paper based the questions will not be there in the final exam. However, there will be some question based on the correlations, and for that, the particular figures will be given to you, particularly when we will be discussing in aeration and agitation. So there are some, let's say, for example, how to read the value of power number. So if power number is not given, you should be able to read from the given figure. I hope this is clear to you, right? Now we can discuss this. Yeah, that's what I'm telling. So this, if such questions are there, then intercept and slope value will be provided to you, and from that you should be able to calculate. Because we understand it will be online exam and you will not have the access to graph paper. So the slope and intercept value will be given if it is required for a given numerical problem. I hope it is clear to you, right? Yeah, thank you. Is there any other query so far you have faced in the assignment questions? If you have any query in particular assignment questions or any doubt related to any course contents or any slides, that also you can discuss here as well. Otherwise, you please raise your questions in the forum that is provided to you. So what I will do, I will quickly go through some of the assignment questions. I hope my screen is visible. So for the week 1, the first question was, what is the net yield of ATP produced at the end of aerobic respiration by one molecule of glucose? So in the aerobic respiration, the first process is glycolysis. It produces 2 ATP. In addition, it also produces 2 NADH. And then NADH is further converted to this ATP through oxidative phosphorylation. So 2 NADH will correspond to 8 ATP. Now further it goes to TCA cycle where one more ATP is produced, one FADH2 and 4 NADH molecules are produced. Now I'm putting this in the bracket because this in glycolysis you get the 2 pyruvate molecules. So in TCA 1 pyruvate molecule is going on. So this number will be multiplied by 2. So this 1 FADH2 will generate how many ATP? And this 1 NADH will generate how many ATP? So if I add 4 NADH, will again generate 8 ATP molecule plus FADH2 will generate 2 ATP molecule. Let me see. So 1 NADH generate 4. So this will be 4 into 4, 16. Yeah, so what it says, this what it says, this for the glycolysis. The glycolysis will be getting this. So there is some technical issue here. Yes, so for glycolysis, we'll be producing 2 ATP plus 2 NADH will correspond to 6 ATP. Total 8 ATP is coming from here. Plus, and in the TCA cycle, we are getting 1 ATP, 1 FADH2 corresponds to 2 ATP plus 4 NADH correspond to 4 into 3, 12 ATP. So this become 12 plus 2, 14, 15 into 2 because glycolysis generate 2 pyruvate molecule and 1 pyruvate molecule generates this 12 plus 2 plus 1, 15 ATP. So this becomes 30 ATP. So in total, you are going to have 38 ATPs. If it is aerobic respiration, which includes this glycolysis followed by the oxidative phosphorylation. There are 8 ATP and 30 ATP, total 38 ATP. Screen is not. Screen is not visible, right? Yeah, can you see my screen? What we are discussing in the glycolysis, we are having. Okay, thank you. Thank you. So in the glycolysis, we are having 2 ATP and 2 NADS. So 1 NADS produces 3 ATP. There will be 6 ATP. Total becomes 8 ATP in getting in the glycolysis. Further, glycolysis generate 2 pyruvate molecule. And in the TCA, 1 pyruvate molecule is generating 1 ATP, 1 FADH2, which is converted to 2 ATP, and 4 NADH, it is converted to 12 ATP. The total becomes 15 ATP from 1 pyruvate molecule. So from 2 pyruvate molecule, it will be 30, and then total 38 ATP is produced by the end of aerobic respiration, where glycolysis and oxidative phosphorylation are taking place. Our next question was, it's very simple based on the serial dilution. So it says this dilution factor is given to you. A number of colony, typically we dilute in such a way that the colony should be countable in the number. So it should be intense, not more than 50 or so. So here we got 10 colonies. So number of colonies is that 9. What was the dilution factor? 10 to the power minus 2. And how much volume of the sample was taken? 100 microliter. That's it. So you'll get 9,000 CFU per ml. This is very simple mathematics. Third question was, what is the Crabtree effect? Crabtree effect explains that increasing the concentration of substrate, particularly glucose, can cause the formation of alcohol under aerobic condition. If you remember, alcohol is produced under anaerobic condition. But if you increase the concentration of glucose too high, then in the aerobic condition also alcohol is produced, which indicates the metabolic regulation is influenced by the high substrate concentration. So it refers to the high glucose concentration as well as the aerobic condition. Question 4 is significance of polycycle in the gluconeogenesis. So it says many times when glucose is not there, still in the biological system, glucose can be generated. Or one example is this lactate to glucose synthesis. So in that case, lactate produced by the muscle will be transformed to glucose by the process of gluconeogenesis. So lactate to glucose. Neogenesis means this regeneration. So glucose is not there and glucose is regenerated from the lactate. That is, and the cycle is known as the Cori cycle. Yeah, so this, if you see the TCA cycle, you will to understand the lysine is not a part of this pyruvate family. The amino acid belonging to pyruvate family are alanine, leucine, and valine. Where amino acid lysine belong to the aspartate family. Which nutrient is essential for ribosome function? So for this, we have already discussed what is the role of micro and macronutrients. So if you remember that, we have discussed what magnesium is essential for ribosomal function, cell wall and cell membrane. Potassium is involved in the carbohydrate metabolism. Calcium acts as a cofactor for enzymes like amylase and proteases. And sodium involved in the ion transport. So the answer is magnesium here. Question 7, which one of the following industrial sector have greater application of bulk microbial enzymes? So for that, we discussed this. In fact, one graph was also provided to see this distribution of enzyme application, and it was detergent sector which utilizes 34% of microbial enzymes. This is next followed by the dairy processes. Which of the following antibiotic is the bacteria by hindering the cell wall? So there are different mechanisms how. Second question in assignment 4. Yeah, we'll come back to it. Yeah. Second question in assignment 4. So this antibiotics, they work by interfering with the cell wall, they interfere with genetic material, or they interfere with the protein synthesis machinery. And bacteria, by hindering the cell wall synthesis, it is the penicillin that disturbs the cell wall biosynthesis. Tetracycline and chlorophenicol, they hinders the protein synthesis, and rifamycin inhibits the RNA synthesis. The next question is where the beta oxidation takes place. We have already discussed. TCA cycle is taking place in mitochondria. Now, yeah, this is very important for the bioprocessing that initially penicillin was produced by Penicillium notatum, but the yield was less. And later on, this even to this Penicillium chrysogenum to improve its yield by many folds. Now we'll go to this assignment 4, question number. Can you explain second question of assignment 4? Now in the assignment 4, this will be before, right? Yes. Maybe visible. Then visual. Second question is please stop sharing. If visual. It's coming. Yes. Okay, when to close it? Share. So far, week 4, the question number 2 is a bioreactor used for antibiotic synthesis. An enzyme has a rate of free soluble rate for fluid soluble enzyme is 10 mole per liter. After immobilization by physical adsorption, the following observations were made. The apparent Km value increased to 0.2 mole per liter, and apparent maximum rate remained the same as R max as 12 mole per liter. Then it is asking what is the percentage of effectiveness factor of immobilized enzyme when substrate concentration is 0.5 mole per liter. So we have to, what is happening here? As of now, we are not seeing any effect of substrate here. Rather, we are seeing the effect of kinetic parameter change in the Km and Vmax. So if I write rate of immobilized enzyme, this will be, let's say we can write Vmax apparent into Cs divided by Km apparent plus Cs. So Vmax apparent, it is given 12. Km apparent is 0.2 and substrate concentration is 0.5. So you'll get this. In case of immobilized enzyme, the rate is 8.57 mole per liter. And in the case of free enzyme, the value is already given. So in the case of free enzyme, the value is 10 mole per liter. So it has decreased from 10 to 8.57, and the percentage will be 85.7%. So this is the rate in the presence of immobilized enzyme and rate in the presence of free energy. So you can see the option D will be correct in this case. Is this clear to you? Is there any other query? Yeah, one more query is for solving the inhibition problem, how to find the intercept value, because I get a different value from the graph when comparing to the answer. Yeah, there may be a slight variation, but there will not be significant change in the answer, because you may be using the graph paper, and so if we are using some Excel sheet to calculate the slope, so there will be some variation in the accuracy, but at the end, that whatever answer will be the closure value to, because the other value, if you see from the option, that will not match with the given slope and intercept value, whatever we will get from the even within the graph paper. But of course, if we are using the Excel sheet or some other software, so those values will be much more accurate as compared to this graph paper. Is there any other query? So the whole idea in this course is to cover the most of this aspects of bioprocess, covering both this upstream and downstream. Then upstream, the kinetics part is important. So this microbial kinetics, enzyme kinetics. Then further, this design of bioreactor operation, particularly in batch, fed batch, and continuous mode. How to do this substrate balance, how to do the product balance. And then further, you can find out how to analyze whether it is a growth-associated product or non-growth-associated product. And at the end, the whole objective in the bioprocess is to optimize the product yield and minimize the unutilized substrate, or you can say maximize the substrate utilization so that the residual substrate becomes zero. However, practically there are some parts which we call that unfermentable sugar. So those remains, but that percentage is not very significant. But the whole idea is whatever fraction of our substrate is fermentable, all should be utilized. And once your upstream is done, the next process comes that how to purify and get your product. So for that, downstream processing we'll be discussing in the second half of this course. Further, when we are discussing bioprocess, then heat and mass transfer are the integral part, particularly for the aerobic bioprocessing. The KLA value is very, very important. So that we have discussed in detail that KLA you can calculate experimentally, and If experimentally it is not possible, then from the given the fluid characteristics, is different correlations we already discussed, you should be able to calculate the mass transfer coefficient. So those, if you will just see the KLA value is also linked with the power requirement per unit volume. So in fact, the addition and agitation, they are very, very important, particularly for aerobic fermentation. Those details we have discussed in detail. So if there is any other query, so please let us know. So if there is no query, then we can conclude. So thank you so much for joining us today. We are going to have next live session. So please share your suggestion, feedback, and comment for the next session. In fact, what will be more productive that please share your question before, and because that link for each live session will be shared with all the registered participants. If you can share, so I can come with better prepared with those particular queries, and we can discuss here in a better way. In addition, this we'd also like, we'll be happy that if you can interact, this orally also. Of course, you can interact through the message and chat box, but you can also unmute yourself and discuss here. So if there is any other comment or feedbacks, here. Otherwise, we are concluding this live session.